This is August 13th, and we were at the International Mycological Congress, and I'm here with Camille Truong and Matt Smith. Camille is from Australia and is in Melbourne. Matt is at the University of Florida. And we've put them here together because Camille had done a postdoc with Matt, and we've all been to Patagonia together and done field work together. One of the things that we do in this is try to get a little bit of a background about what people have done and how they've come into mycology. And I know, Camille, you have a very complicated pathway into diverse pathway into mycology. Why don't we start with you?
Yes, of course. So I did my PhD on lichens, systematics, and at that time I fell in love with Latin America and the Andes, and so my PhD was about describing new species in the genus Usnea in the tropical Andes. And so after that, for my postdoc, I wanted to do something on mycology, but also explore new horizon. And that's how I ended up in the lab of Matthew Smith for a postdoc on ectomycorrhizal fungi in the temperate forest of Patagonia.
I thought I remembered that you did some plant taxonomy or plant ecology earlier.
Yes, so for my master, I did a study on population genetics of mountain birch, so Betula, in the north of Europe. But I think that was mostly because I studied in a rather small university in Switzerland, and at these places there were no mycology possible. So I remember I already had an interest, but there were no offer about it. So that's actually how I ended up doing this, which was good. I learned, lab work and statistics, good. So it was useful.
And Matt, how about you? How'd you get started?
Great question. Yeah, I became a mycologist totally accidentally, which I think is probably the case for many of us. I always thought fungi were interesting, but I took a class as an undergraduate when I was at University of California at Davis, and it's a school that has plant pathology, so they have a mycology class, and I needed a lab course. I was a biology student. I was interested already in evolution and ecology, but I just happened to have mycology fit in my schedule, and I fell in love with mycology pretty quickly, and a big part of that was that I really loved it when I asked questions to the instructors, the professors who taught the course, that quite often the answer would be, well, we think the answer is, and then they would give what they thought it was, but it was clear that that was kind of a hypothesis or a speculation sometimes. And so I thought, well, they seem really smart and they know a lot about these things and they still don't know the answer. So, maybe I can make a contribution to this field. So that's kind of how I got hooked. Yeah. And then I was really interested in the tropics. I worked as a technician in the tropical forest in Panama, and really liked that a lot. And so I think I was always attracted to the diversity, the high diversity, and the novelty of just so many organismal interactions. And mycology is good for that, but you can do it in your backyard. So.
You didn't quite do that with your PhD, though.
No, well, my PhD, I ended up working on ectomycorrhizal fungi for my PhD. In oak forests. So I was in California, and then I left California and came back for my PhD there. And I also did my PhD at UC Davis in Dave Rizzo's lab. And I worked on ectomycorrhizal fungi, and I also got really interested in hypogeous fungi there, because it turns out there's a lot of diversity of Pisizales and hypogeous fungi in a lot of these more Mediterranean woodlands where I worked.
So what were your findings from that PhD?
Good question. Well, I looked at temporal change in ectomycorrhizal communities. So it was before next generation sequencing, before high throughput sequencing. So I think for my PhD, I generated something like 2,500 DNA sequences, which seemed amazing at the time, but is not very many now. But I sampled over time and found that actually spatial orientation of where the samples were taken was more important than time in structuring those communities. And then the other thing was that it was pretty early on in being able to figure out what the things were from roots was challenging. So the approach that I used was to do this cloning and sequencing. So we had pieces of DNA that you could get ITS and LSU from the same piece, and that really helped to try to identify especially what a lot of the, for example, Pisces aeles fungi at that time, there were no reference sequences, and so figuring them out was what was actually ectomycorrhizal was part of the problem. And then I also worked on the systematics of some of the truffles. So I worked with Jim Trappy was kind enough to sort of take me on the side to mentor me in truffles. So that's how I got interested in those. Yeah, hooked on those.
Camille, you ended up at one point in Tierra del Fuego. That's pretty amazing.
Yes, this is a wonderful place. I left my heart in Tierra del Fuego.
Me too.
You too.
What did you do there?
So my story with Tierra del Fuego started I was traveling there, and I saw the forest it's a angiosperm tree called the Notophagus, which is the dominant tree there. There is no ectomycorrhizal conifers in South America, in Patagonia. And I could see it was in the fall, so some of the species are deciduous, so they had these like beautiful colors. And I could see the structure of the forest because there is another species which is evergreen, and it really makes this line of it's green and orange and then green again, for example. And so that's how, when I saw this, I had started to be interested a little bit in community ecology, and I was reading some things, and I really saw this pattern, and I was like, wow, that would be a really cool place to do research. And at that time, I think I heard that Matthew Smith was getting large funding to do some work in Patagonia. We hadn't, have we met before?
I think we just met at a meeting.
We met, but yeah, we
briefly.
didn't know each other, and I had never done work with ectomycorrhizae before, so I remember I contacted him, and he told me, write a proposal, and then we can talk. And I remember that, so I was back to Switzerland at that time. I had options to do, in Switzerland they have very good fellowship for young scientists to do postdocs abroad. And so I remember that I spent an entire month reading and writing a proposal, which was pretty new topic to me. And then I sent it to Matt, and it was, this is how it all started. It's how it all started. So the idea of the project was to look at the ectomycorrhizal soil community in these different forests and see how are the different communities according to these different hosts. Some of them are deciduous, some of them are evergreen, and they really form monodominant forests. It's very highly structured. Or if it has to do with the soil type, for example, and especially the kind of nutrient that these trees, like the nutrient requirement of the different trees. And we actually find really interesting patterns because one of the tree species, which is called Notophagus pumilo, who is a deciduous tree, had like the typical organic nitrogen economy of these, cortinarius ectomycorrhizae that enhance nitrogen limitation, for example, which was very similar to what you see in coniferous boreal forest, while the evergreen tree had kind of this nitrogen, was growing on this nitrogen-rich soil, like you see in the temperate rainforest of Canada, for example. So it's very, very different nitrogen economies. And it's really interesting because it's kind of shifting paradigms from what we know about, boreal zone and all this idea we have about how ectomycorrhizal function. And now that I'm in Australia, I see this all the time. Sometimes things are very different in the southern hemisphere. So
Yeah.
that's why, it's really exciting to do research there because you have to just forget about what you know and start over.
Great. I feel like we're all family here in a way. And part of that is that with Patagonia and southern South America, that Matt and I did a trip when he was a postdoc
Yes.
in, what, 2000?
2008, I think.
Was it
Yeah, I think it was
2008?
2008.
Yeah.
Yeah.
And that was kind of the first time that we'd really gotten into some of these fungi.
Yeah, that was my first time in the southern hemisphere. It was great. It opened my eyes to how different it was.
Yeah, we'll talk about that difference a little bit and what you
Well,
think you've found.
Yeah, so I was a postdoc in Don's lab, and he said, "Oh, I'm going to go to Patagonia for 11 days. Do you want to go?" Yeah, I think I would like to go. That sounds really fun. And so, yeah, we went to two different places that were quite different from each other. But especially, we went in part because Don had the transcribed diary of Roland Thaxter, and we kind of wanted to go back to some of the places where he had written about. And I think the coolest thing about that was that we went back to the places that he went back to, and we found some of the same fungi that he had drawings of and specimens of, and at least some of them were not described yet, which I just thought, wow.
And it
there's
was
so much work
100
to
years
do.
later.
It was 100 years.
Oh, well, exactly 100 years.
Not exactly, but close.
Pretty close, yeah,
Wow.
pretty
Yeah.
close. And so, yeah, we ended up describing a Rulandiella that he had clearly described in his diary and had pickled collections in the Farlow herbarium, and now we've described it. So that's pretty exciting. But yeah, no, there are so many. It's quite different, like you say, the southern hemisphere. And I think for me, so I've studied ectomycorrhizae a lot, and one of the things that's very distinct about southern South America is that it does have this really almost complete dominance by one plant group. And that's really unusual. There aren't really other places in the world that have really just one ectomycorrhizal plant group like that. And so it's kind of a unique opportunity to study someplace quite different from other places that have ectomycorrhizal plants and fungi.
And Camille, you're now in Australia.
Yes, so it actually made the connection to Australia because while I was doing my postdoc, we started to look a little bit about the biogeography of some of these groups, and we very quickly realized that sometimes the most closely related species were species from Australia. So we collaborated a little bit with researchers there as well, and so when the position opened there, it was kind of a logical way for me to go from like to go across the globe, but still on the same hemisphere, because Notophagus is also present in Australia. It's a very different situation actually in Australia, because Notophagus there, apart from maybe in Tasmania, it's really a remnant tree of past climate. In the state of Victoria, it only grows in like this really tiny, small ravine, which are protected from fire. It's probably heavily endangered by climate change, ultimately. And yeah, I think in terms of biodiversity of ectomycorrhizal fungi, Australia is just the craziest place in the planet, because there we have all these Gondwanan fungi, but then there is also all the sclerophyll. So sclerophyll forest dominated by eucalyptus. People don't always realize it, but there's more than 1,000 species of eucalyptus, 500 of acacia, and then a lot of other host trees. So the diversity of host trees is enormous, and then the diversity of fungi is just enormous in Australia, way more actually than in Patagonia. Yeah.
Why don't you talk a little bit about your jobs now? Matt?
I just talked.
Yeah, sure. I am a professor at the University of Florida in the Department of Plant Pathology. I have an interesting job with an officially I have a 3-way appointment that includes research, obviously, and teaching. I get to teach fungal biology. And then I also have an official extension appointment, which kind of includes a bunch of things, including identifying fungi for poison control and the plant disease clinic. And also I'm the curator for the fungus collection at the Florida Museum of Natural History. And I feel very lucky because I get to study basically whatever I want if I can get enough money to do it. So that's an awesome place to be lucky to study fungi. And then yeah, I just became a full professor not too long ago, so I'm probably not leaving anytime soon. I'm enjoying it there.
Great. And graduate students?
Yeah, we have well, right now we have 2 graduate students, but yeah, I've had graduate students working on a number of different topics. I've had some graduate students work on zygomycete fungi with Gerald Benny, who works with my group and is emeritus, and he's now 82, so he doesn't work as much as he did, but he still loves fungi, and he's been a great collaborator. And then, yeah, I've had folks working on truffles, bird's nest fungi, bird fungi interactions, so a bunch of different things. That's one of the things that I like about my job, is that I get to learn a lot. So I try to do different things all the time.
Yeah. Camille, you have a very different job.
Yes, so I'm a research scientist at the Royal Botanic Gardens Victoria in Melbourne. So we have botanical gardens with the horticulture department, but we also hold probably the largest fungi collection in Australia, the Mel Herbarium. And so my position is a little bit in between research and curation. I have to say that we have really good curation staff, so I don't have to do a lot of basic curation things. They're very good at digitization and all the things, but we support them, and obviously we try to use the herbarium for research, describing new species. There's a lot of collections that have been done in Australia, but not so much species descriptions. So there's hundreds of species that just lay in the herbarium waiting for descriptions. So that's part of my position. And then I'm also honorary associate at the University of Melbourne, so I can co-supervise students in University of Melbourne, but actually also other university. We have at least 5 or 6 different universities in Melbourne. So I actually have quite a lot of students that I co-supervise, because I guess we offer a very special setting at the botanical gardens that maybe the universities, they don't have, like mycologists doing systematics. So I have 3 PhD students that I co-supervise, 2 masters and 1 honors, which is like a bachelor in the US maybe. And then I guess the last part of my job is to do assessment, and also maybe we help the local governments to take decisions, for example, about conservation. Me and my colleague Tomé, we also responsible for emergency poison calls. So whenever there is a poisoning case, we have a phone 24/7 that we have to answer. So yeah, like Matt was saying, it's quite diverse, and this is something that I really like about mycology. I really like field work, but I discovered while I was studying that I actually really like lab work as well. So and then we can do culturing, and so it's been quite good, and interesting. I've been there 2.5 years now, and so hopefully I can stay now and establish a little more, which is really exciting.
So both of you have been in these interesting habitats to do collecting and to think about projects and so forth. From each of you, what's a really cool fungus that you've encountered?
Ooh, what's a really cool fungus? It's very hard to choose the coolest fungus. I think I'll just choose one that stands out in my mind, which is Guyana-gaster necrorhizis, because it's just it's a very weird thing. It's related to armillaria, but it's a truffle-like fungus. And I remember the first time that I saw it in the field, It just doesn't seem like it should be real. It's
Where did you see it?
in Guyana, so it occurs in Guyana, and it's got a dark outer peridium that kind of looks like a tuber melanosporum or one of the dark truffles, but then it has a little stipe and it's always connected to decaying roots. And when you cut through it, it's very hard to cut through it. It's so gelatinized that when it dries out, it's almost impossible to cut. And the first time I saw it was actually from an herbarium specimen before it was described. And I got DNA sequences back from it, and they were armillaria. So I figured I had screwed something up in the lab and contaminated somehow from armillaria, because it just doesn't look anything like armillaria. It seems totally unlikely that it would be related to that. But then I got to see it in the field, and it's when it's mature, it's like bright orange, weird gelatinous thing. So that's I guess off the top of my head, that was a pretty cool,
good choice.
Pretty cool
good
fungus,
choice.
yeah. Pretty big mystery what it was when we
Yeah.
found it. So
Yeah. And Camille?
But it's good. Now it gives me time to think about it while you were talking.
there's so many. Yeah.
Yeah, there's a lot. I think the first one that came to the top of my head, and I think it's a good one because it kind of relates to the connections that we have, is the genus Notojaphnia. So Notojaphnia was described by Pfister when he went to Patagonia, and it's a cup fungus, Ascomycete ectomycorrhizal fungus, which I think is really interesting because people don't necessarily think about Ascomycete when they think about ectomycorrhizal fungi. It's in the Tuberaceae, which is also interesting because people usually think about truffles when they think about Tuberaceae, but it's not a truffle, it's a cup. And yeah, so I guess it's also a southern hemisphere group, which kind of linked my history with Patagonia with now my present, and my future in Australia. And I'm actually working on the group at the moment because in Australia, so there's one species that was described in Patagonia and one species described in Australia, but in Australia we have more diversity than that. So I guess, and it has pretty nice spores as well. Ascomycetes have really nice spores, so that's why I like them. Yeah, so yeah.
That's a good choice. That's a good one, definitely.
Well,
A cup fungus in a truffle group. That's weird. Yeah.
Described by Pfister.
Yeah.
yeah, it is a really cool fungus, and I remember that Matt sent me the specimen when he was trying to figure this out, and I thought, that's what that is.
Yeah.
So,
Yeah.
Notojaphnia, yeah.
Yeah,
Good.
and also interestingly in Australia we find it in like this recently burned sites as well. So it seems that they have interesting ecology as well. So
Great. Well, thank you both for coming out and doing this. And I didn't say who I was at the beginning, but I'm Don Pfister, and I've spent my career at Harvard University. So thank you.
Yeah. Thank
thank
you.
you.